Hello, everyone, and welcome. Thank you for joining us for today's session, the sixth in our new webinar series titled Environmental Monitoring Considerations for Gene-Drive Modified Mosquitoes. My name is Justin Overcash, and I'm with the Foundation for the National Institutes of Health, where I work as part of the Gene Convene Global Collaborative. Our mission is to support and advance best practices and informed decision-making for the development of genetic biocontrol technologies to improve public health. To do this, we work to identify and address key questions, provide technical advice, and strengthen capacity and information sharing. An important part of the testing pathway for new genetic vector tools will be post-release monitoring for safety and efficacy. This webinar series will highlight current vector and biodiversity monitoring efforts across Africa and introduce potential methodologies and tools that could be used to support environmental safety monitoring following release of gene-drive modified mosquitoes, which are being developed for malaria control. Sessions will take place and have taken place weekly through the first week of June. Before we get started, we have a few housekeeping reminders. Please email events@fnih.org if you have any technical issues. Your video and audio will be off during the webinar. The presentation will be between 45 and 60 minutes. During the presentation, feel free to submit questions anytime in the chat. Questions will be addressed at the conclusion of the presentation. All webinar recordings will be posted to the Gene Convene Virtual Institute and the Gene Convene Virtual Institute YouTube channel. Today's webinar is titled A Chance to Measure Life on Land, the Promise and Challenges of Airborne eDNA. This is the first of two webinars in a series that will describe the potential of eDNA as a tool for tracking biodiversity. And we are honored to be joined by Dr. Elizabeth Clare. Professor Elizabeth Clare is a globally recognized expert in biodiversity science with a research portfolio that spans molecular ecology, environmental genomics, and the innovative use of DNA-based monitoring technologies. She completed her PhD at the University of Guelph in Canada and held a prestigious NSERC postdoctoral fellowship at the University of Bristol and Cardiff University in the UK. From 2013 to 2021, she served as a faculty member at the Queen Mary University of London before joining the Department of Biology at York University in Toronto, where she currently leads the Clare Lab. Professor Clare is a Fellow of the Higher Education Academy, a Fellow of Linnean Society of London, a Research Associate of the Royal Ontario Museum, and an Honorary Fellow of the University College London. In 2024, she was elected to the Royal Society of Canada's Scholars, Artists, and Scientists, one of the country's highest academic honors. Her lab is widely known for pioneering the use of DNA barcoding to study the diets of predators, pollinators, and parasites, and more recently, for groundbreaking work demonstrating that environmental eDNA can be recovered from air to assess terrestrial biodiversity. She is now at the forefront of international efforts to scale biodiversity monitoring using air filtering techniques integrated with national air quality infrastructure. Professor Clare's work has been featured in top-tier journals such as Science, Nature, and Current Biology, and has reached wide audiences through media coverage, podcasts, and educational outreach, and has even played a role in finding the solution to a murder mystery. Very interesting. She is also a creator and host of Bio Audio, a freely available podcast series that serves as a learning resource for students and the public, which can be found on Apple and Spotify. In addition to her academic contributions, she serves as Associate Editor for both Biological Invasions and Environmental DNA. Before we get started, a quick reminder, I'll be moderating the Q&A session following the presentations, so please feel free to post your questions in the Zoom chat at any time. Additionally, a reminder that today's webinar is being recorded and will be posted to the Gene Convene YouTube channel after the event. With that, I'll turn it over to our speaker, Dr. Clare. If you can go ahead and share your slides, we can get started.
Okay, are we sharing now?
I can see them.
Excellent. Okay, well, thank you very much for the invitation. I love talking about biodiversity and DNA, but I'm going to start with an error that I sort of ran into a few years ago. I was being interviewed on the radio, and somebody asked me to begin with, why is biodiversity important? And to me, it's such an obvious question and answer that I made a complete mess of the question. And I said something about bees and agriculture, and I kind of passed it off. So I now start every talk with that question, just so I can get it right. So why is biodiversity important? And for some people who will be watching this, like me, it's just obvious, but for many, it's not. So I'll go through my quick list of why you should care about why we're doing this. Um, the first is that thing that I sort of tried to answer when I was posed this question, which is food security. There's tremendous amount of data that suggests that biodiversity is directly linked to our productivity. The biodiversity of soils is directly linked to our ability to produce nutritious food above ground. So this is an obvious link. You cannot feed a planet of our population if you haven't got biodiversity to support that productivity. More subtly, people don't think about water in the same context, but water purity is directly linked to biodiversity of things like marshes. And biological systems are still one of the best ways to actually purify water. Plants are very good at doing things like removing contaminants and pollutants from our water system. And so things like marshlands and the biodiversity of marshlands are still really, really important in providing the free service of water purification. More interesting in this day and age is the regulatory effect of biodiversity on climate. There's some really interesting research from Montreal in Canada from a colleague of mine showing that increasing the biodiversity of tree cover in a city can actually lower the temperature by 2 or 3 degrees, just having more diverse trees. And so if you think about climate change and 2 or 3 degrees being the sort of threshold that we're currently concerned about, we can actually mitigate some of that effect just by increasing tree biomass and biodiversity in urban spaces. Erosion, there's some classical ecological examples of rebalancing ecosystems, things like the return of wolves to parks and the tremendous effect it had on regulating biodiversity at multiple different trophic levels. More interesting from a business point of view, pharmaceutical development and bioprospecting generally rely upon finding new areas of biodiversity that have not been quantified previously. The UN has a huge report out about the link between cultural practices on a global scale and biodiversity, and the use of indigenous knowledge in trying to better understand the cycles and history of biodiversity. Art is largely inspired by biodiversity. In Canada, where I am, some of our most famous artists were inspired by our natural landscapes and biodiversity. Human health is directly linked to our exposure to more complex green spaces. There's some really interesting studies now that look at things both about your physical health and your mental health, that both of these things can be greatly improved simply by increased exposure to more complex green space. It's not enough just to go outside. You need to go to places that have a more complex environment to experience them. And this can regulate both your physical and mental health. So biodiversity is actually important to all of us on a very individual level on multiple aspects, even if we're not thinking about it particularly commonly. So what is biodiversity? Well, we can start by talking about it in different contexts, and it's a really hard question to answer. So if I was talking about biomass, so the actual amount of matter on planet Earth, then I might say that biodiversity is largely dominated by worms and arthropods, so all the insects and things like that. Worms below ground and arthropods above ground. That's largely the biomass of terrestrial biodiversity. If I was actually trying to quantify this at the level of what we know about life, the things we've counted, the vast majority of the names that we use to describe species are in the arthropods. So insects are the most well-known biodiverse, most number of species we've counted group out there. But that doesn't really reflect necessarily what we know about biodiversity. So on a biomass point of view, it's lots of worms. On a names point of view, we've described a lot of insects. But if we took up the information that we actually collect, and here's a really good example that was taken from a news article a couple years ago. GBIF is one of the online tools for tracking biodiversity information. And there was this running joke for a while that it's basically a database about birds, that most of what we collect in terms of observational data is about stuff we like to follow and things like bird watching. If you use iNaturalist or NextBird, some of these online tools, they're massively dominated by people's personal observations, which tend to be things you see commonly like birds, which led someone to write in Forbes this article that said ugly species deserve protection too. And one of my friends describes these little things as the little things that make ecosystems function. We know very, very little about the small species that actually drive all the ecological processes we care about. Those things like producing biomass, producing productivity above ground, purifying water, it's recycling nutrients through our ecosystems. They're all done largely by small species that we don't know that much about. And a few years ago, there was a really interesting demonstration done by the Guardian newspaper when I was living in England, which says, okay, well, if we don't know much about biodiversity, what have we done to it? What has the impact of our very existence as humans been on this thing that we need but don't know much about? And the answer they sort of came up with, they tried to quantify, was that since the rise of human civilization, we've lost individuals at a very, very surprising rate. So about 83% of individual wild mammals have been lost, not the species, the actual individuals. If you went and counted how many zebra there are and how many frogs and how many fish and how many plants, it's the individuals that we have currently lost. We've rebalanced the distribution of where life is. And probably the most dramatic example of this is if we look at mammals. There's about 6,500 known species of mammal. So I've went and counted them all up, the cows and the pigs and the bunny rabbits and the giraffes and the elephants. There's about 6,500 known species. But humans and our livestock represent roughly 96% of the individuals. So if you were an alien and you came to Earth and asked, what are the mammals like? You'd say, well, Earth is dominated by people and cows. And only about 4% of the actual individuals are what we call wildlife, the other 6,455 species. And the same thing you can see if you look at birds. There's about 13,000 known species of bird. 70% of the individuals are domestic wildflowers. So we're a planet basically of cows and chickens and people. That's largely what individual life is dominated by. It's only about 30% of the birds are actually what we call wildlife that you would go out and try and find if you were a bird watcher. So this is what we sort of are talking about when we talk about a biodiversity crisis. We may not have lost all that many species, but what we've done to the biomass distribution and abundance is the real drama, that the world is basically dominated by you and me, cows, dogs, and chickens, and that everything else has become a very, very minor component of what we call biodiversity. So what is the biodiversity problem then? Well, what most people are really shocked to discover is that about 90 to 95% of the species that live on Earth are completely unknown to science. We have never seen them. They have no name. If you caught one and you brought it into a taxonomist, they could not tell you what it is. They could probably tell you what family or genus it belongs to, but there is no name for that thing. So I'm a taxonomist. I've described species. And I tell my class this, I always say, if you want to describe a new species, it's really, really easy. Most things have never been described. Go out and find one and describe it. It's not a hard thing to find a new species. Why is it so unknown? Why do we know so much about so little? Is that we don't, as a planet, have any infrastructure for measuring biodiversity. We just don't do it. We do biodiversity assessments using taxon-specific tools. So I go out to study my favorite animal locally in one habitat where I work. I go every year down to Belize as a part of a team, and we study bats in this one area of Belize. And then we try to extrapolate up what that means for the rest of the country or the rest of the neotropics or the rest of the planet. So we take these very, very specific local studies and we try to bunch them together to generate a picture of what biodiversity is and how it is changing. But no scientist would ever plan an experiment that way. You don't do that largely. So what's the problem here? The problem is that biodiversity has no standardized approaches, very, very little. So the way I study birds, I don't learn about other mammals at the same time or other bats. If I go to study bats, I won't learn about bunny rabbits or birds. If I go to study a frog, I won't learn about fish. You learn about only the thing that you've set out to study. And that's really the challenge of biodiversity science. We don't have any large scale way of doing this. We can't go to the top and look down and measure everything at once. That just doesn't exist in this science. So that's the problem of biodiversity. That's the challenge that we as biodiversity scientists have. Most of it is unknown. It's tremendously changed in the way it is distributed amongst the world, and we don't really have any good tools to go and measure it at large. We're really, really good at intensive small-scale studies. We're really bad at doing that at any sort of large scale or replicative method. We just don't do it that way. So we're going to change tracks now and talk about environmental DNA. So what is it? It's not actually particularly new. It was talked about as early as the 80s in the context of some microbial work. It then kind of disappeared for a while, and it came back in the late 90s, thanks to a group in England led by a man named Eske and a couple of his colleagues who began looking in things like this. This is an ice core. And what they discovered is that all organisms, you, me, the tree, your pet hamster, we are all shedding biological material all the time. We leave it behind like a fingerprint behind us, like a footprint. It's bits of dead cells, fragments of hair, even bits of saliva while I'm talking will become lost from us, and they carry with us genetic components. We call all of this material collectively environmental DNA, or eDNA for short. It's just a way of discussing bits of stuff you leave behind, and you can't stop it. It just always happens. All organisms do this all the time. And what Eske and his colleagues discovered is that you could cover it and you could use it. And he went looking in ice cores like this one to see if he could find DNA from ancient life. And it worked surprisingly well. He found material that he was able to link back to things like mammoths from, thousands of years ago. He went and looked at mud cores, and he found extinct moa DNA. He found bison. He found herbaceous woody plant material. He was able to reconstruct extinct ecosystems based on DNA that had been trapped in ice or sediment cores for up to 100, 200,000 years. So he showed that basically this DNA was lost to the environment. It could be used to reconstruct ecosystems, and it could persist for a long time under certain circumstances. This has quickly expanded to become a huge field of research, particularly in the last 15 years, really in aquatic areas. So this is one of my students, Alejandro. He just finished his PhD, and he was using DNA in water to survey for remnant and older habitats that have been replaced by invasive species. So in his case, he was looking for the axolotl. This is the famous little animal on the screen, famous in the pet trade. This is the little salamander that never really emerges. It stays aquatic its whole life. People love to keep them in fish tanks. They may be extinct in the wild. They actually haven't been seen in a long time. There are a few refuges that they may still persist in, but we don't catch them anymore. And so he went looking for the traces of DNA to see if he could find evidence that they still persisted in the wild. We don't know the answer to this. They may be on the verge of extinction in the wild. We don't know. And so this is him taking samples in and around Mexico City, the habitat of the axolotl he was after, and trying to find if there was any remnant DNA left that would indicate that this critically endangered species of axolotl still persisted. And this is where eDNA has really had its forte in aquatic systems. We can use it to take a scoop of water and survey an aquatic population of fish. Traditionally, we would use things like electrofishing or seine netting, which are much more invasive. Here, we're just looking for a scoop of water, and that water contains within it DNA of all the organisms that have gone through. And it's become so popular that we now look for eDNA everywhere. The picture in the top middle is actually taken, I think, from the USGS, just showing you the sort of approach to surveying fish populations from a scoop of water. But we can find eDNA trapped in soil profiles. In honey, you can get the DNA of both the plants and the insects to show the ecological interaction that created the honey. In rainwater washed off leaves, in insects that feed on us, so a mosquito feeding on a mammal, it has eDNA of that mammal in its gut. You can track where it's been, and even left behind in footprints, for a famous example of taking DNA from footprints in the snow. And so it's found in this really wide variety of places. Now, we've used eDNA in a lot of really interesting contexts, and this is one of my favorite. This is blood meals of terrestrial leeches. So I had a student, Rosie Drinkwater, who went out to Borneo as part of a huge team surveying terrestrial diversity in forests, and she did it by collecting leeches. And so she would go out and these are in the leaf litter, and if you stand still long enough, they're going to come and find you. She scoops up hundreds or thousands of leeches, and you can get the blood meal DNA from their gut. And they basically become an army of little field assistants that have been visiting the different terrestrial vertebrates and recording their presence. And so by sequencing vertebrate DNA from the gut of a leech, she was able to survey the mammals in particular in this forest setting. So from leeches, she found things like elephants and sun bears, DNA from bearded pigs, and interestingly, the pangolin at the bottom middle, that's one of the most trafficked animals in the world. Had I sent her out to Southeast Asia and said, please find and track some pangolins, she probably would have failed. But I sent her out to find leeches, and she came back with pangolin DNA. And so this is sort of the power of environmental DNA. It accumulates in really unusual places, like the gut of an otherwise fairly common leech. You can use the leeches to find evidence of the mammals, and it becomes like a bit of a forensic system where you're tracking indirect evidence for species that you couldn't necessarily find. She got the idea of using dung beetles, these famous beetles that roll around mammal dung in balls. This is some footage provided to me by my friend Rob Nell. And they push around this dung, and in doing so, they get coated in the DNA of the animal that left the feces behind. Dung beetles are super easy to collect, easier than the leeches. You build a really simple dung beetle trap. That's what's in the top right picture there. You dig a hole and you suspend some feces in a little pot, and the beetles are pulled in by the smell and they fall in the pit. And then you can scoop them out. You can swab or take all the material off their shells and sequence it, and you find evidence of what mammal dung they have interacted with. That tells you the mammal had been there recently. And she did this sort of study, and she found things like banded civets, the same bearded pigs. And so another army of field assistants that are out there collecting the material for us at really good rates. So we were doing this kind of work in Southeast Asia, looking for new sources of environmental DNA. And now I'm going to ask you to play a little game. This works really well when we're in person because I can get us all to do this at once. But I'm going to ask you, wherever you are watching, to just take a deep breath. And I promise that in 20 minutes, when I'm finished, you will never think about that deep breath the same way again. So take a collective deep breath for me, and I'm going to trust that you're playing along at home. Around the time that we were working with leeches and dung beetles, I was commissioned by a group from the chief scientist in the UK to write a report on how we could use DNA to understand terrestrial ecosystems, to really measure biodiversity as part of the 25-year strategy of the British government to start incorporating DNA at a very large rate into their understanding of ecosystem resilience and how to preserve biodiversity on national scales. And so I was writing this report about all the different sources of DNA, and I sort of summarized what I just showed you. We could collect it from soils and rivers and all of these places on land. And I said, and of course, we should filter the air. And when you write a government report, you usually have to give a case study. And so I collected all the case studies for all the different ways you can find DNA. And I just assumed that we had filtered air for environmental DNA the same way we filter water. And it turned out we really hadn't. As recently as 2021, there were effectively 4 papers published on the topic. There's a huge body of literature on things like pathogens, so how viruses and bacteria travel through the air. But at the time, we didn't really think about anything else. I found basically 3 stories. That it was primarily theoretical, that we should look in air, but no one really had. There was 1 or 2 papers that looked at plant material, and that's not a terrible surprise. If you've ever had seasonal allergies, you know that plant material floats in the air. And there was 1 story that I found about fungi. I think it was in Hawaii, that there was fungal spores found in the air. Again, not a shock. But really nobody had looked to see whether other forms of DNA floated around with us. And so we devised a really simple experiment to see if we could prove that eDNA moved in air. At the university where I was at the time, we had these colonies of naked mole rats. And this is right around the time that COVID really became a big problem. And we were suddenly very restricted in what we were allowed to do and move and where we were allowed to go in the UK. And one of the things I could do was go to the animal care facility. So these are naked mole rats. They live in captivity. This is some footage from a friend of mine who's an expert in naked mole rats. And so we devised this experiment thinking if eDNA was ever going to build up in the air, where we would detect it would be places where animals lived in close contact in small environments, and then we would get a sort of concentration of DNA could be there. And so we basically took the same pumping system that we used to take water out of a river and filter DNA, and I stuck it in the naked mole rat enclosure. And so what you can see here is the mole rat sort of crawling around. And then this is a little external nest box chamber that we could pull the air out, and we filtered it. And our assumption was that we would get naked mole rat DNA in the colony and that the room would be empty. We were wrong in the most spectacular way. Yes, there was naked mole rat DNA in the colony, but there were lots of other DNA sources everywhere. The room itself was full of DNA, human DNA, mole rat DNA, even dog DNA, which it turned out the person who feeds the mole rats was tracking DNA in because he walked his mother's dog on the weekend. And so on Monday morning, when he came in, he would track the dog's DNA into the room. It would become aerosolized, and we were capturing it with our filters. So we were wrong in thinking that the room was the empty space. In fact, it was full of DNA signals. And some of them we've tracked back to different sources subsequently. So we were wrong, but for really good reasons. Does animal DNA travel in air? Absolutely. It was all over the place. So we decided to see whether this would work outside. And again, we were really restricted where we could go. Within my allowed travel distance in England, where I could go, was a place called Hamerton Zoological Park. It was shut down because of COVID, but the animals still had to be fed and cared for. And so I got permission to basically walk around the zoo for months by myself, taking air samples. In order for eDNA and air to be useful at all, I've been thinking about burrow systems and subterranean life. We would need to be able to collect it outside in the actual environment. And so I had this idea that if I knew the map of the zoo and where all the animals were, and then I was able to basically remap it in air samples, could I create the same map again? And zoos are wonderful because you know where everything is. They're all non-native species, which are confined to certain specific areas. So we know where they should be on the map. What does DNA tell me? And so I walked around the zoo taking samples inside enclosures and outside in their habitat where they can walk around. And this is just a sort of top-down view of the zoo. And there's the little red circles are my little sampling points for some of my data. And what I'm showing you here in these boxes are that if I am outside the meerkat enclosure, I can detect meerkat DNA. So that's what I've detected in that box. And if I'm over with the dingoes, we get dingo DNA. And if I'm in the primate house, we get lemurs and howler monkeys and tamarins and sloths. The stuff that's there, we can detect it. And this is good news. It tells me that I'm confirming the map. But we also discovered other stuff. So over in the gibbon enclosure, I also found donkey DNA from the enclosure beside them. In the Tyra enclosure, there's lemurs and binturongs and dingoes and budgies. This is DNA on the move. This is air currents moving DNA short distances through the zoo. And this is actually really good news. Because if I'm standing in front of the tiger and I detect the tiger, there was no point in doing the molecular work. I can see the tiger. But if I can detect it hundreds of meters or kilometers away, now I have a useful tool. I can detect something that I can't see. Now it becomes very, very interesting as a method to survey large areas. So what we saw here was that DNA moved. It moved with wind currents short distances through this zoological park. So the strongest signals were always from the animal that was there, but there were other signals as well from other parts of the zoo that were being moved. And we also found some wildlife. If you are in England and you just find hedgehogs, people get really, really excited because hedgehogs are endangered. And we had hedgehog DNA wandering around. This is an open park. The animals can walk through. So squirrels and ducks, muntjac deer are invasive locally. They're actually from Asia, but they're locally invasive in the UK. They've been released from old estates. And so we're seeing wildlife signals in the zoo. And this is animals that are able to walk back and forth through the open parts of the zoo and their DNA actually as well. So this was really good news. This tells us that we are able to actually survey an area using nothing but a filtered air sample. So the next sort of question we had was, well, what is it telling us? So we know that in the very earliest eDNA work, they were looking at 400,000-year-old signals. If that's what I'm measuring, it's of very little interest because I need to know what was there now. I don't want to know what was there 50, 60 years ago. And so we wanted to know whether it's really reflecting recent activity or really old activity that just sort of builds up. And so we went back to actually the people who'd surveyed plants in Texas, and they contacted us, and they had looked for plant material on sort of 2-week scales over the course of a year, but they'd never thought to look for animal material. So they asked us if we would go back and look at their DNA and see if there was animal material in the same samples. So they have these wonderful little dust collectors. These are basically a metal tube with a fin on the back, and they will always turn into the wind and spin around. So the wind blows in and blows material into the little collectors. You come by every 2 weeks and you wash it out, and that material now becomes your biological sample. And we went looking for mammals and some birds. And this is sort of what we got. What you're looking at here, one of the only data slides I'll show you. Each column is a 2-week period over the course of a year. So there's 22 2-week periods that we measured for. Each horizontal is a different species, and the green squares are where we detected them, and the gray squares are where we didn't detect them. And so the first major pattern is that there's this period in the first sort of half of the year. Well, there's one other pattern first, which is that this is a bunny rabbit. Common stuff is common. So the bunny rabbit, you almost always detect it because there's lots of bunny rabbits. This is a really rare thing, a Texas toad. We only detected it once. It's fairly rare. It only emerges under very specific conditions. And so a rare thing is rare. This is good. It means we're not just accumulating stuff in the environment. But the biggest pattern is that this is empty. This first period of the year where we sampled, very, very little was detected. This corresponds to the time in Texas where it's really hot, there's really reduced vegetation, and a lot of animals actually leave this area. You get very, very low biodiversity when the environmental conditions are unfavorable. And this is reflected in what we can detect in the air. So few animals leads to very few detections in air. And this is actually good. This tells us that we're not going to detect stuff when it isn't really there. In contrast, this time of the year is when Texas gets cooler, rainy, windy. It's actually mating season for most of these animals. So they come back in, they get very, very active, and suddenly our detections begin to go up. So we're suddenly seeing the animals' activity on the landscape. This is great. This tells us these signals are relatively recent. If they just accumulate over time, this should just be one big green grid. I don't want that. What I want to see is that when an animal is present, I detect them, and when they're not, I don't. And probably the most dramatic case of this is actually that Texas toad. The Texas toad only emerges from this really dry environment when there's huge rain activity. There's an ephemeral pond on the area, and they come and they sort of come out and they get very active, and then the pond goes away and they disappear. And the week that we detected this, the 2-week interval, there was a massive rainstorm in the middle of it. So we were able to actually link that airborne DNA signal to a known environmental event that caused the emergence of a frog or a toad. So this is really good. This is telling me that the signal is being induced by a known environmental behavior. So this was really good news for this. Now, we then wanted to move into larger, more complex communities, and I work with bats. So every year I go down to Belize. We work on a large scale survey for bats in Belize, and most of those animals are brought back very temporarily for an hour or two into one location where actually it's my job to identify them all. So that's my job on this trip, is I'm one of the few people who can identify all the bats. So each of those little white bags you're looking at has a single bat inside, and it's waiting for me to go through and quickly tell you what it is before we release it, or somebody might collect some sort of material from it, or some measurement that they're taking. So my job is quickly sort. And what you're seeing on the other side here are some handmade, basically tiny vacuums. We build these. They are really simple, little tiny, low-scale vacuums based on fans. And on top is a filter that can collect biological material. So we designed these, and if you look closely under those bat bags, you can see 3 of them lined up on the shelf behind. And so we deployed these in the room to ask the really simple question: had I not looked at all those bats and named them all, could the DNA signals in the air have told me the same information? How good would I be at telling you the diversity of this room where the bats came? Think of it like a cave. These bats had flown in and flown out over the course of time. Would those 3 samplers sitting there have been able to tell you who came and went from the room? We were astonishingly successful. We were about 95% successful at identifying and documenting every species of bat that came in and out of that room using just the airborne sample. And I'm the backup. I know what was there. So I can tell you how accurate we were. I think we missed 2 in the course of 2 weeks, 2 species. So an incredibly accurate way of surveying space. Now, we began deploying this now at large. This is one of my students, Nina Garrett. She's actually sitting inside a tunnel cut by looters into a Mayan ruin. And so this is some of the oldest architecture, oldest engineering you can find in the area. It's probably a 2,000-year-old building. It's had this tunnel carved in it, and the tunnel is now used by bats. She's setting up the newest in engineering. These are tiny little environmental DNA vacuums. And so she sets these up in as many roosts as she can to try and document the ecology of roosting of bats without having ever had to see the bats. So we deploy them inside these sort of artificial caves cut by people, but also some other areas. Tree roosts are a great place. That tree on the left there, the giant Guanacaste tree, it's probably got 500 different bats from a dozen different species inside of it. Can we document that? And so we leave behind these little air filters to just gradually cycle the air through a filter over the course of a night to find out who was there. We were really, really successful with this. So bats that are commonly found in these roosts were recovered with their DNA. Here's 4 examples. We know these bats are in these roosts because we survey them using things like nets and camera traps. We already know this. So you're looking at here's a fruit bat, an insectivore, a nectar bat, and at the bottom right is a vampire bat. All of these are commonly found in these roosts, and we recover them with DNA. The really interesting thing was the DNA did a better job than us. It has now twice found extremely convincing evidence of the presence of another bat, which we have not caught in 20 years of surveys. It makes sense for it to be there, but they're really good at evading nets, but they couldn't evade the DNA collectors. And we have now twice documented them in tree roosts. In 25 years of working in the tropics, I've never seen one alive. So the DNA is getting there before the people at this point in our ability to say they're probably here, but we are missing them from our inventory. And this has happened a couple times now. This is another type of roost where we actually drop the samplers down into it. We can't enter it. You need rappelling gear to get down into this. Is looking down through the hillside into a very deep crevice that goes down quite a ways. We are lowering these little vacuums into it because we can't get there. This is not really surveyable by us. And in one of these, one year we documented the presence of a bat called Chiroderma, which we, again, never caught there. We'd actually dismissed this as some sort of error in the data, except the next year we caught it there. The bat there is Chiroderma. So the DNA predicted the presence of a bat that we had not caught, and then we caught it the next year, demonstrating the DNA had been right. So the DNA predicted correctly the presence of a species that we had not physically seen before, and then we confirmed it. So that's a wonderful sort of validation of this as a method. So we know that animal DNA and plant DNA are carried in the air. You can collect them through things like dust collectors that spun around in Texas, or we can actively collect it through these little handheld vacuums that we can build. And it's reflecting recent activity. It's giving us accurate information about who's who and where they are in the landscape. We've now trialed this in some other contexts. This is a very modified version we've used for surveying beehives. We're interested in diagnosing honeybee health and foraging dynamics. This is a really, really safe little, we call it the Bee Safe sampler. It's a very, very tiny vacuum about the size of your baby finger, and we can put it in a beehive and we can document the health and behavior of those bees just through air that's being cycled out of the colony. This is all really, really fun. These are great new tools for surveying diversity, but it doesn't solve that initial problem. This is actually a photograph of my first field site when I was a PhD student in Guanacaste in Costa Rica. And, wonderful new tools we've developed in the years since I was there, but we are absolutely no better at telling you who lives in this forest. We don't have a way of just going out and surveying this kind of a landscape. This is a great little thing for beehives and bat roosts. It's really fun and it's really accurate and it's great for doing surveys, but it still doesn't scale. And this is a problem. The problem we've always had with biodiversity, that we don't have a standardized approach, because of that, we don't really have standardized metrics for measuring or quantifying biodiversity because we don't have a way of scaling this stuff to global infrastructure. And some of the largest agreements on biodiversity, things like the Convention on Biological Diversity and the UN SDGs, the Nagoya protocols, they all say that at national scales, countries are responsible for measuring, quantifying, and tracking biodiversity, and not a single country in the world can do it. They do not have the tools to do it at that scale. We're left with people like me and a plot of land that I can survey for one thing I'm interested in. So how do we bridge this gap? And this is what we were fundamentally stopped by. How do you fix this? And it turns out there is a solution. And the solution is so elegant that I'm having more fun as a scientist now than I think I've ever had. We wrote a paper about the zoo, about surveying Hamerton Zoo Park. And it was read by a physicist. He actually read an article written by the Natural History Museum in London about the paper, and he got really interested, and he wrote a letter to my collaborator, Joanne Littlefair, and I, And he said, I don't really understand biodiversity. Can you explain the problem? And so we gave him the 2-minute version of what I just told you, that biodiversity is fundamentally changing at unbelievably rapid rates, but we lack any ability to quantify it. We can't really measure this on the scales we have to. And then we told him about air sampling and what we were doing with bat caves and beehives. And he said, so you just want to filter air on large scales? And we said, well, yeah, but I could make a million little handheld vacuums and deploy them on all the trees in the forest, but someone has to maintain them, and they're electrical, need batteries and all this kind of problem. And he said, well, I work on air quality. Have you thought about using the planet's array of air quality sensors? And we'd never heard of them. It turns out many countries in the world, probably most of them, have some level of network like this picture from the United Kingdom. This gentleman, he actually works, James Allerton, he worked for the Heavy Metal Detection Network. This is a network of air quality samplers set up very specifically for decades ago to measure our exposure to things like lead and arsenic, stuff that is a byproduct of the burning of fossil fuels, because they have some level of risk to your respiratory health. There are legal limits in most countries on what you can be exposed to, which means we have to monitor your exposure. So these sampling systems are set up in grids across many countries, and all they do is filter the air, looking for tiny particulates in very specific size ranges so that they can assess your exposure to these kind of potentially harmful pollutants. The sample precisely, this particular brand, 1 cubic meter of air per hour, and they run continuously, and they're basically automated. Every week, a new filter like the one on the top is moved in. It runs for a week, filtering particulate matter. When it becomes effectively saturated, like the little piece you can see at the bottom, it turns brown with all the material it's collected. It gets shifted out, and a new one comes in. And James said to us, it's just sampling particulates from the air. Is it possible we have accidentally been sampling biodiversity for literally decades? And we were so focused on the physical particles, we didn't think about the biological ones. And we said, we don't know, but we can test that. So we set up this little interesting experiment with the Heavy Metal Network with James and one of his colleagues, and they sent us some samples. So one set were from the Scottish Heathlands. This is a fairly rural area outside Edinburgh. This is one of the sort of typical sampling stations as part of their network. It's just operating as normal, taking weather. This is a weather monitoring station. It helps you feed into weather networks. It's taking particulate matter samples. It's measuring all kinds of environmental variables in this sort of rural area. And then they had an experimental place where they actually developed these kind of technologies right in the middle of London. This is beside a place called Bushy Park, which is basically an urban forest in the center of London. And they sent us some of these. They basically, from those two locations, they sent us only 17 little triangular fragments of filter paper. And they sent them to us. They had nothing interesting happened to them. They gave them to us, and they asked us to see if we could sequence or find biological material on those fragments. They were unbelievably successful. From only 17 little pieces of paper, we found and recorded the presence of 180 different taxonomic groups, dozens of plants, birds, insects, mammals, fungi, amphibians, spiders, even a couple of fish from the area where we think there's a big bunch of sand that gets aerosolized. So this was unbelievable for a couple of reasons. Number one, they already exist. There's no infrastructure needed to build. This stuff is sampling all the time continuously and has been for decades. And the second was the diversity of things they were recording. Remember that most of our biodiversity sampling techniques are specific to the animal or plant you are looking for. When I go looking for bats, I don't learn about birds at the same time. In this sense, we can learn about basically everything all at once. And we were only limited by what we looked for. So we started looking for plants, we found plants. We started looking for fungal DNA, we found fungal DNA. So the more things we looked for, the more things we found, preserved on these little tiny fragments of paper that were quite literally being thrown away because they're done, they've been used. And so if we look at this, I'm going to show you one data slide, and I'm going to take away most of the data to make it really simple to understand it. But it's really important for a couple of reasons. So this is all the data from those stations, and I'm going to get rid of most of it. These three boxes are the plant material. And what we did at one of these experimental sites was we looked at what would accumulate in an hour if we took the sample away after an hour, after a day, and after a week. And if you are a scientist and you're looking at this, those are actually not statistically different. The same number of species basically recorded between a day and a week. And we puzzled over this for a long time. Why weren't we getting more species of plant the more we sampled? And then, of course, it occurred to us plants don't move. We'd basically saturated, we'd measured everything in the available area. This is actually really good. It means that we aren't just accumulating stuff that blows in from somewhere else, in which case it should keep going up. If we have an end to sampling, it means that the wind is not overly affecting this. This is really the local area, and we finished sampling it. We're done. In contrast, animals, here's the yellow my little vertebrates thing, the longer you sample, the more you find, because basically more birds come over the course of a week, more and more things visit the area, and you catch more and more kinds of DNA. So it's really different than the plants where they're just there and you can sample them quite quickly. With animals, the more that turn up, the more things you sample. And that's really good. And the second thing that came out of this is these samples I'll show you. These ones came from Scotland, and they're also the same basically as London. You get the same number of things accumulating, more or less. They're really important because what we didn't know was these were not fresh samples. The ones they mailed us from Scotland were a year old. They had sat on a shelf for a year, and the DNA didn't degrade. There are countries that have archived these samples. They don't throw them away. Some countries throw them away and some save them. What this tells us is that even those really old samples may have usable DNA that has not degraded. It'll depend how they were stored. But here's a good example where a year later, we can still recover the same information. We haven't really lost any. It tells us there could be absolutely archives of DNA out there we can use. And we've begun to do that. We had a paper actually accepted this week, and there's another that we've submitted for review, where we've gone back and actually done what we said we should do, and we've surveyed the entire country. Each of those plots on the map is one of these heavy metal detection network stations. They gave us all of those little filter papers going back for an entire year. We targeted different taxonomic groups with different genetic markers to see whether it was possible for us to basically survey an entire country. And the answer was yes, we can do it. I'm going to not show you all the details of it. I'll show you this, which is meant to impress you with the amount of data we have on birds and mammals, other vertebrates, arthropods, other invertebrates, protists, fungi, plants, and then the one that's really interesting, stuff that shouldn't be in the UK. And those are not errors. They're the most important data in an entire national survey. So we tracked 28 orders of life, more than 1,000 genera for a year, and it cost us zero in terms of fieldwork. This is simply done from the garbage of an existing piece of infrastructure where they would have thrown the data away. And we were able to survey 28 orders of life across the entire country. But why is that not in the UK stuff the most important? Because one of the big questions is, where does the DNA come from? At first, you get worried when you find a Cape water buffalo being detected in Scotland, until you Google it and you discover there's a herd there being raised. Or you find some interesting wildcat, and then you realize that the sampling station is right next to a zoo. These are real detections of things that are not native to the UK, parrots that live in central London in effectively the wild. These are really important because we know where they came from. We know the exact location of the zoo, of the herd of water buffalo, of the parrot colony in London. Once you know where the DNA came from and where it was detected, you know how far it traveled, which gives us the very first ever estimate of how far this DNA can move, which tells us basically what we are sampling from each of these stations. And once you know that, you can build a range map. So this is actually the first range maps for species in the UK built using nothing but air samples, having never seen the animals. We have overlaid the air sampling, the red dots, which tell us where we detected the species, against black dots, which actually come from citizen scientist observation databases, and the gray patterns, which is what their known distribution is. So we're now trying to map, based on the distance that we know that these stations can collect from, what we detect versus what people report from citizen science databases from what the government knows as the potential range of the species. So it suggests that we can actually, in the future, this is very preliminary ways of doing this, use this type of air sampling at high density to actually develop range maps and track the movement of populations. The last thing I'll show you about this is that we can start looking at the effect of things like urbanization. So because these sampling stations are also measuring information about environment, things like pollution levels, disturbance levels, weather, and we know where they are, we can begin to look at the effects of different gradients of anthropogenic human activity on diversity. And what you're seeing here is just all the birds on the top right, all the mammals on the top left, all the mammals on the top right, the plants and the fungus. And we've separated those sampling stations into the ones that are associated with human population centers, so high levels of human impact, and those that are associated with very much more rural areas without a great deal of human impact. And what I want you to only notice from this is the overlap between green and blue in the different grids. So if you're a mammal and you live in the green areas, the really rural areas, versus mammals that live in the really blue areas, the urban areas, there's basically no overlap in those taxonomic communities. They're very different communities of animals. If you're a fungus, they're almost completely overlapping. There seems to be no real effect on fungi diversity of the gradient of human effects. But if you're a vertebrate, like a bird or a mammal, there's a huge effect on the community composition. So we're able to actually look at where the species are from our detections and how that relates to what we know about the landscape. And it seems like we can here now look at the impact of human activity on the community level in ecology. So why is this my proposed solution to that taxonomic problem? Remember the problem of we don't have a way of measuring at scale. And the reality is this is not a UK only solution. Here are two maps. This is a sampling station like the one in the UK that we use. The map on the top right there is Europe, and every one of those blue dots is a sampling station, just like that one, measuring almost exactly the same way over and over and over again. The map on the bottom is the US. That's their grid of samplers that we think measure exactly the same way. And those two, as far as we can tell, are measuring about every 3 days, over and over and over. This is literally millions of biodiversity samples being collected that no one had recognized before. They're basically thrown out after you analyze the pollution components to it. But what we have here is infrastructure across huge portions of the planet that are capable of measuring life on land. And these are easy to build as maps because the language wasn't a barrier and it's publicly available. But I have also found them in places like New Zealand, Peru, Brazil, South Africa, China. They are found in many parts of the world. They're concentrated in the global north, but they are not missing from the global south. And these are the kind of things that we can use to actually take snapshots of biodiversity at the scale of countries and possibly continents. There are many versions of these measurement tools that exist out there, but if you turn your phone on and go to your weather app, it will give you an air quality index. It's probably been generated from a station just like these. And you can get that kind of data from almost anywhere in the world, which tells us that there are sampling stations out there collecting very similar kinds of material. Not all of them save it. They don't all preserve the sample, but the ones that do are accidentally measuring biodiversity at the same time, which means we have the possibility of tracking biodiversity on global scales. I'm going to stop there. There's, of course, lots of detail in that I'm happy to answer as questions of there's lots of challenges to this, but it presents a real solution to how you could measure biodiversity at the level of an entire planet. And I will give some acknowledgement to my funders. Thank you very much.
Thank you very much, Dr. Clare. That was a fantastic talk, and I see we already have tons of questions rolling in, so I'm going to go ahead and get started. The first question was from Tovi, and the question, well, fascinating. What is the area of collection you believe you could collect from? Which I think that question came in before you started showing continental U.S. and Europe and talking about those points. But he had a few more questions on here. Is it quantitative or only qualitative? Is there an approach to move these data from species lists to community compositions?
Those are fantastic questions. So the first one, what is the range? This will depend slightly on what model you're using. So my little handheld version measures, you need 3 of them inside one cave to accurately document the cave. That's our current estimate. That will depend on basically how much air you're moving across a surface and how big your surface is. Those seem to be the 2 most important criteria. How big is the sampler and how much air does it move in a given time period? So we've now got some estimates that, if you're doing a bat cave of a reasonable size, you need 3 or 4 samplers distributed. And as the cave gets bigger, you need more of them. So that's one scale of measurement. Now, the scale of measurement of these networks of air quality sampling systems, our estimate there, we have a sort of a minimum and a maximum, and the average is something like 25 kilometers. I put that in some very, very enormous confidence intervals because those are the very first estimates anybody has ever made. And again, it will depend on your method. So these ones are incredibly regulated. They were set up by physicists and chemists to measure something very specific, and based around particulate motion models. Is that right? Particulate motion models. So they know precisely what they are collecting, and we think we're collecting about the same thing. And that looks to be borne out mostly by those rare detections of stuff that we could trace to a location. But this is where we get into a bit of arm wavy stuff. So my guess at the moment is somewhere between 10 and 70 kilometers, which is actually a fairly sweet spot for measuring this. If you want to find an invasive species and you can draw a circle on a map that's about 50 kilometers wide, that's a really nice target for a good survey of whatever the habitat is. Had it been 500 meters, I'd get really worried because you'd need millions of these things to do the job. If it had been all of Europe, it's less interesting because you can't narrow down where it's from. You can learn something on a temporal scale, but not on a spatial scale. So that actually you need multiple different scales. One of my colleagues in Sweden is doing something similar with a different sampling system that is doing really large scales, which gives really fascinating temporal data. So it comes a little bit to the question you're asking, but that's our sort of vague realm of estimate. Really big, powerful ones, continental scales. This sort of network system one where there's way more of them are smaller scales. If you want to do a beehive, we can do one beehive and it's different than the beehive next to it.
Very cool.
Quantitative. Is it quantitative? This is the most dangerous question in environmental DNA work. So there are many, the simple answer is no, but with some hesitations on that. There are different ways you can attempt to quantify environmental DNA. They are very, very risky. I didn't show you a data slide about that, which we could look at if you want to, but there's so many things that can throw off those measurements. It becomes tricky. The power is not in an individual sample you could quantify. The way I often talk about it is if you're looking at astrophotography, okay, so a photograph of the night sky, you could try and take one photograph for 2 hours, and if someone jiggles your camera, you destroy it. What most astrophotographers do is they take 1,000 photographs about 10 seconds long and they stack them all together. Each one of those photographs is fairly meaningless. It's got 1 or 2 little points of light, but when you stack them together, you find the Milky Way. That's what we have here. What we have is hundreds of sampling points taking hundreds of samples over and over and over and over again. Any one sample has very little in it and is meaningless. But when you stack hundreds of samples together, you end up with a snapshot of a country. That's the power, is that this is one of the few biodiversity sampling systems I've ever seen that is truly spatially, temporally replicated. It's taking the same measurement over and over and over again in the same way in multiple different locations. That's the power, is replication. You can begin to see an animal that was always there and disappears, and we don't see it anymore. Where did it go? The new invasive species that suddenly appears. You could track the range shift with climate change. Those are the things that you have the potential of tracking this way.
Very cool. Yeah, I imagine that the number of samples is super important. Let's see. Our next question is, and you answered this in your talk. Did you find any insect DNA in the filters? Maybe you could speak to. I guess one of the questions I had was, right, when I think of eDNA, right, mammals are probably shedding tons of DNA, but then insects might be shedding less. So is there anything to maybe the mass of insect DNA, or does it matter because you're
yeah, I think I can go back here. So there's the arthropods and other invertebrates. I think, to give you a vague estimate on that, of the 1,000, more than 1,000 genera, 700 genera were insects. So yes, there's tons of insect DNA. Now, compared to the actual biodiversity of insects, that's probably not that high. There's way more in the UK. You need more samples and more sequencing to always find more. There are certain groups that are harder to detect. This is a terrible method if you want to follow one thing in particular, because I can't direct it. The whole point is that I can't target it. It's a generalized system for monitoring life at scale. If you want to follow your favorite bird, you're way better to go out and look for it with people. If you want to measure birds, this is a good way of doing it. So it comes down slightly to the question you're asking. This is about how we measure all of life in one way at a very, very broad scale. I could never narrow it down and find my favorite bat this way. You can't track one thing. So it's a different scale of question than we've ever asked before. And that's a sort of important differentiation here, is that this is about tracking large scale biodiversity at large scales.
Right. So, and that's interesting, right? Because when we think about environmental monitoring, I know at least from the risk assessment standpoint, you can have general surveillance, and then can have case specific surveillance. So, if we're talking about a gene drive mosquito, and we wanted to keep track of, say, general surveillance of and biodiversity in an area, I think the airborne eDNA would be very effective and would kind
Yes,
of keep the baseline.
and if your mosquito suddenly disappears or reappears, that's the sort of information you might find. We've always measured it here and now it's not there anymore, or we have never measured it and suddenly it's appeared. This is like baseline data from which you start measuring aberrations.
Right, right. All right. Let's see. Oh, we have a lot more questions, so I'm going to keep moving. This is from Moez. Incredible talk. What are your views on how eDNA may be utilized to monitor release of genetically engineered organisms in selected areas? For example, considering the release of a gene drive mosquito.
Yeah, so it's a really interesting question. I get this question frequently. Usually a different context. It's something like, I'm studying this particular bumblebee. Is there a way we can survey just for it? There are complementary technologies like qPCR, which can be targeted to much smaller things and get you some abundance data, or a specific species assay. We don't that's not the area that I work on, but people develop those. I reviewed a paper recently about assay development for a species of interest. You can absolutely do that. And it's one of those things you begin to layer the technologies together. So you do a mass survey, you have the right genus of mosquito in the area, and now you layer on a species specific assay or a gene specific assay. There's people who work on things like eRNA for looking at functional genes in environments rather than the taxon themselves, interested in things like ecological processes. You might look for sugar metabolism as opposed to bees, or you might look for nitrogen cycling genes that are responsible for that. So you can absolutely layer on technologies. You know that the general ecosystem is like this with eDNA, and then you layer on a quantitative assay for your species of interest and an RNA assay for the gene of interest. You can begin building these things up as layered, sort of hierarchical technologies.
Very cool. And I know in my mind, I'm thinking, all right, maybe you could come up with an RNA assay for Cas9 or something like that. It's part of the gene drive construct, so you could detect yes. presence or absence, which I think would be very useful. Our next question is from Bill. Some countries have selected protecting biodiversity as a protection goal in environmental risk assessments. Can this goal be practically quantified and implemented, or is it too broad of a goal to monitor?
I guess it depends on what you mean by that. It's a very broad statement. Yes, I think it's attainable, absolutely. And there's things we know will protect biodiversity. For instance, protecting habitat is the number one thing about protecting the biodiversity. Larger, more intact pieces of habitat are better at preserving biodiversity. It doesn't require any scientific measurement to know that. Where this type of technology, I think, is incredibly useful is sort of three things: detecting the new invasive that could potentially be damaging. We're already doing that. You can use it to monitor the status quo. Is it changing? And if so, how? And the third one that people are beginning to do is trying to quantify interventions. So there's lots of interesting things, trying to add things like corridors between habitats for movement. Has anybody quantified whether that corridor is being used? Can you actually measure that? And there's many ways of doing that. You can measure in the corridor. You can measure the diversity of the populations on either side and see if they're exchanging alleles. Is this newly restored habitat actually supporting the life we expect? Are we restoring the communities in terms of the different trophic levels that we know need to be there? And we deal with that in bats a lot, because lots of bats are some of the first in the tropics, the first things that actually do things like disperse seeds. They get there before the birds do a lot of the time, and actually are primary dispersers of some of the pioneer plants that move into habitats that are being restored. So there's different layers to that question of what you mean by protecting biodiversity. And some of them you can answer with DNA and some of them you can't. None of us advocate for the replacement of techniques, but we're trying to fill this gap, which we can't really address with current techniques, which is how do you measure at the scale we actually, a lot of the times, have agreed to measure in these sort of agreements, but can't functionally do.
Right. All right, let's see. The next question is from Stephanie. Can you clarify whether samples from air quality sensors can give us information on trends in abundance of species? And do these sensor networks exist in Africa? I know you mentioned South Africa, and you've hinted at the quantitative versus qualitative question a little bit.
Yeah, the simple answer is, can you get quantitative data? Yes, but it is to be treated with a great deal of caution, I think is, yeah. And like, I don't bother, because I know how difficult it can be. You start with some of the things that you can't even control. Shedding rates. You mentioned that with insects. Do insects shed more or less? Well, it depends on the insect. My main collaborator on this, Joanne, refers to it as the flappiness factor. If you've got an animal like a bird that flaps continuously, it's you might propose it's going to lose more DNA than something that doesn't move like that. Something like a newt or a snake, which doesn't produce plumes of bits of feathers and stuff flying around. So there may be biological predictors of who sheds more. And if you're just picking up shed DNA, now you've got a problem, because you don't necessarily know those factors. So that's before you even get to the lab-based things, that primers have biases and techniques have biases, and DNA degrades at different rates, and was the animal dead or alive, and all kinds of interesting problems that crop into it that make quantification hard. That being said, there are quantitative assays, particularly when you target them at a small group, that you can develop. So there seems to be, even beyond all that, if you just took the amount of DNA and looked at, there seems to be a relationship between DNA and biomass. But that relationship is subject to very, very constant changes if the wind blows through, in this sort of sense. If it rains, my suspicion is you basically washed everything out of the air and you start over again. But I don't know that. It's just a suspicion. In the classroom where we first quantified this, I can show you the picture, which I'd skip over it unless we want to talk about it. This is a quantification of the number of species along the bottom and the amount of DNA we actually sequenced from them. And there is actually a correlation. As you get more individuals, so more abundance of a particular species, they produce more DNA in the sequencing reaction. But notice that there are 4 major outliers to this. There's 3 at the top, where there's way more DNA than there should be. And there's 1 at the bottom called Desmodus rotundus, where there's way less. The explanation for this took us almost to the proofs of the paper to figure out. And the answer was me. I caused this inadvertently. Remember, I'm the person who identifies the bats. The ones at the top are the hardest to identify. I spent more time looking at them. Desmodus rotundus is a vampire bat. I can tell it instantly. So my action as a taxonomist, looking at the data I was there to look at, influenced my own sequence recovery.
So they were literally, you had them out of the bags longer, so you collected more?
Yes. Or we had to open the bag more than once, or, so for instance, with Glossophaga mutica, I have to examine the front incisors to correctly identify it from another Glossophaga that is there. With Corolla perspiciolata, I have to measure tibias. And I can do that quickly, but not as quickly as other things. So this is me as basically the problem in my own data. So if I can have that level of influence, I begin to worry about other things that are having influence that I can't quantify. We've got some other cases where we know why there was a sudden plume of DNA in the air. Somebody cleaned the room and literally re-aerosolized things. So there's so many ways that you can actually alter that distribution. There's Nina in a cave. The act of going inside that tunnel will have produced dust. We don't know the impact that has or how long it lasts. So I don't quantify because I can't know with any certainty about what that quantification can mean. So there is a relationship, but it's so easy to throw it off. Most of us don't do it.
Right. A lot of variables that could impact the amount of DNA that you're collecting.
But there's also some really interesting new technologies coming on that may help us get around that. What we need is sort of massively parallel qPCR, which is beginning to be produced in different systems.
Very cool. All right, let's see. I know we have quite a few more questions. Let's move on to the next one. And this is another one from Toby. Based on the number of species you listed, it seems that after a week, only a fraction of the true biodiversity is detected. What are the key biases and sensitivity limits?
That's a very good question, and at this point, we don't know very many of them. So, on some level, saying that we've recovered more than 1,000 different genera across the UK is probably the largest scale biodiversity inventory ever done at once, and also a teeny fraction of what's actually there. So, obviously, it's either both really good and really bad at the same time. We don't know a lot of the factors that currently predict what we will or won't detect. And that's true of other groups that have worked on this. I stood in front of an animal and took an air sample and didn't detect it. And I don't know why. It was right there, I saw it. So why didn't I? And yet some other animal that I never saw is one of the most abundant signals. Why? These are things we don't know. And that's why most of us don't quantify. What factors are important in detections? My guess is abundance, activity, and physical composition of that animal. Or, well, true with plants too. If a plant's pollinating, it's probably going to produce a huge amount more biological material than if it's dormant. Here in Canada, I would predict you don't get much plant DNA in the winter and lots of it in the spring, but I've never measured that to check. So there's lots of things we can predict that will have an effect. Now, it's true, 180 taxa in a week's worth of sampling in a location is not everything that was there. It's also way more than any other survey method would have detected. And so that's both the good and bad part of the story, is that it's still not perfect, but it's this gap space where we don't have any other approach. In that work in the UK, the national survey that we've published, we detected stuff like protists, non fruiting body producing fungi, that there is really no good way to monitor for those. We got lots of them. So it's also getting life that you wouldn't normally pick up on a traditional survey. And we've done that kind of quantification in the paper and looked at, what does citizen science inventories find versus what we find. And they're almost non-overlapping systems, because we're not looking specifically for charismatic things and they're not finding invisible things like spores. So this is not a replacement system, but a highly complementary system in this sort of space where we don't have a system.
Right. Our next question is from Maiga, who does some monitoring work himself. He said, impressive and thank you. How available are the filters that you used? Where can we purchase them? Do you have any advice?
You mean the little handheld ones, I assume? We build them. My kids build them on the dining room table. I've even got a video on YouTube you can watch on how to make your own. So we just make them. They're made out of computer fans, and the filter material is actually the same material you put in your furnace. So it's a very high grade MERV type filter that's graded down to viral size particles. We've experimented with different sizes, so you can build them. At the moment, there's nowhere you can buy them. I'd love if somebody wants to. I've actually tried to get companies to build them, and no one wants to do it. But I tend to build them and mail them to people just so they can use them and tell me how they work. So yeah, they're relatively easy to make. My nieces and nephews can make them. So if you're interested, you can always send me an email and I'll direct you to the video on YouTube on how to build one.
Very cool. Our next question is from Anusa. Thanks, Professor, for this nice presentation. Maybe I've missed something, but what can you say about the accuracy of airborne eDNA in identifying the species present in the ecosystem? How is this type of sample preserved?
So the accuracy will depend on what gene you target and what database you have. So there's a sort of dual problem. DNA degrades and degrades quickly. So we target small regions, but that limits the information. So, for example, the reason I quote the number more than 1,000 genera is because for the 700 genera of insect, we did not try to do species level identifications. I don't think many of them are right, but the information we're getting is so small that there's an error in the identification. And the second, I'm much more confident with things like mammals because there just aren't that many of them in the UK. I'm pretty sure we got the right ones. The second problem is the databases themselves are hugely incomplete because we only know about 90% of the life on Earth. So if I went to somewhere, my favorite field sites in Central America, and did the same insect survey, I'd probably be limited much to families and some genera of stuff that's commonly occurring elsewhere because the databases aren't as complete. The actual how you assign any sort of designation is it will depend on where you are and what you're trying to assign. If it's a mammal, we'll do really well. If it's a vertebrate in general, probably pretty good. If it's an insect, most taxonomists can't identify most insects because most insects have never been identified. So it becomes a missing data problem still. There are ways you can still quantify. You could count the number of unique genetic signatures to try and quantify an area, even if you don't know what the DNA actually is. You can say, I've got all of this, all this huge diversity of bumblebee DNA here and very little here. This habitat probably has more bumblebees in it, even if I don't know what they are yet. And you can then, we could try and refine those identifications in different ways. So there's a sort of a two-based problem, which is the degradation rate of the material and trying to find a marker that preserves enough information without missing taxa because it's too degraded, and the database you're using to identify, which in itself is a fragmented thing with lots of misidentifications in it.
Very good. Let's see. We have, looks like 2 more questions this one's from Calvin. What is the approach you use to process the air eDNA? Say you have your sample of air, what do you do next? So a little bit more about the processing.
Okay, so the process is like vacuuming. So the whatever your suction device is, it's pulling air through a filter. So I often say it's like making coffee. So if you're making coffee, you've got a filter that traps the grounds and the water goes through. It's exactly the same process. We're pulling the air through and the particulate matter gets stuck. Now you have a filter with particulate matter on it. And just looking around my desk if I've got one sitting here. I usually have one folded up. So you've got this little piece of folded material and it's trapping some particulate matter. What we do is basically we cut that piece that was exposed out of the material. Now it's basically a tissue sample. So we can take a little piece and put it directly into like a QIAGEN DNA extraction. Or if we've got larger filter samples, we actually we soak in a buffer and agitate the sample for 24 hours to basically knock the material off and then use the liquid phase to concentrate the sample to get it into the QIAGEN extraction. But the material becomes the equivalent of a tissue sample. It goes in your QIAGEN extraction and you go through a regular DNA extraction procedure, and now it's just a DNA sample. Now you can target whatever you want. Primers for fungal life, plant life, and we've got bat-specific primers, we've got more general vertebrate primers, whatever it is you're interested in, you can target from that sample. And we're trying to add more and more regions. It becomes a cost-benefit analysis of adding more genes and more targets and based on what you're trying to measure.
Very cool. Let's see, our last question, or maybe it might be 2 questions from Tovi. I wonder what is the cost of this analysis, which given the number of samples, it looks like most small labs may not have the budget for that. And if you require a central facility of sorts, what do you think? So like what's the scope of the laboratory needed to process the samples? And then the second question was, have you considered using drones to collect the air samples or use them already? What are the limits and potential benefits?
Those are really good questions. And I realize I also missed the question about how the samples are preserved.
Oh, yes.
The best preservation method will be freezing. And the second best is dry and in the dark. So most of our samples get frozen as soon as we can. And the ones from the air monitoring stations, they usually, because it's part of their procedure, go in a petri dish, get sealed and stacked on a shelf in the dark. And that seemed to work for at least months. There is some degradation, but it's not disaster. We've now got them putting them in the freezer. So it's, we're trying to slightly change their operational outputs to try and make them more accessible for biological outputs. So the question about cost of the analysis, it's very difficult to estimate. It is substantially less than fieldwork, way less. So there is a definite savings if what you're trying to do is large scale inventories. If you're trying to find your favorite bird or your favorite bee, you're probably better off with one person who can do it efficiently in the field, and you're done. But at the level of trying to survey lots of different life, it's much, much less expensive than field work. And you can do it more rapidly. So Nina Garrett in the picture here, she can monitor dozens of caves at once for bats because she can just walk around and drop the samplers off rather than having to stand there herself operating the net all night long and doing one cave. So there is a time efficiency here as well. In terms of the actual cost, this is not as much as people think because there is an economy of scale. So in fact, the more samples you have, the cost per sample goes down. You can throw, we put 4,000 PCR products on our last sequencing run, which was a year's worth of samples times 5 regions times 3 technical replicate PCRs that had been pooled. So there are massive economies of doing more material. One sample is very expensive. 1,000 is not that much more expensive than one because there's a fixed cost at the end, which is usually the sequencer. That cost doesn't change. So when you got that in the equation, PCRs and extractions are actually relatively inexpensive in some cases now, depending on your facility. Now, dealing with the scale of the problem of potentially hundreds of thousands of samples, you need a factory. And there are commercial labs that do this now. There's a couple of commercial partners we've worked with that are capable of processing environmental DNA. They are usually more expensive than the average lab because where they're usually set up for high scale diagnostics of one sample, there aren't any that I know of that are actually dealing with the scale at this point, which is a very different approach to how you manage material in a lab. The bigger challenge, I think, is actually that most labs can't handle environmental DNA, and most environmental DNA labs can't handle airborne DNA because of how these are forensically tiny signals. They are so easy to contaminate. There's a running joke that what I consider clean is now a totally different thing. I asked you to take a deep breath about half an hour ago. And there's 2 reasons to think about that. Well, number one is you're never going to think about a breath the same way again. If you go out and wander around in nature and you take a deep breath, you are basically a biodiversity sampler. Your lungs are doing the same thing these things are doing. It's a much more interestingly personal connection to nature when you think about the fact that you are inhaling it. It's part of you. I kind of like that idea now. When I walk the dog, I think about that. On the other side, when you breathe out, you just contaminate all of your samples. So that's the scale of the problem. You produce more DNA than you possibly collected on that sample filter. So the chances of you destroying your own sample is high. So what we're really using is effectively ancient lab type facilities that take us out of the equation as much as possible. Trying to think of the other questions. What sort of facilities? So what we need is an industrial scale ancient lab. Such things sort of exist. But that was what would actually be needed if we wanted to operationalize this as a truly continental level survey method. On the other hand, my little lab did a country, one postdoc. So the concept that one person over the course of a year could conduct an interestingly reasonable level national survey, that's pretty impressive in terms of manpower and what it costs.
Sure is.
Drones. No, I don't use drones. There's 2 reasons. They're fun, but they have 3 problems. One is there's lots of places banning drones. You need a license in a lot of countries to operate a drone. So, I can't get a license to use one in most places I want to work. The second problem is you still need an operator. It doesn't take the field work out of the equation. One person has to send that drone out, and you need a person standing there as long as the drone is operating. It actually isn't a cost-effective process. If you're going to do that, you might as well be doing traditional on-the-ground field surveys. It doesn't help in that sense. So and it doesn't monitor for as long. The systems we're using, we set them out, we walk away. You come back a few days later and collect your material. There's no hanging around watching it. Nina leaves these behind in the tree roost, walks away, and the next morning collects them, and the data's been collected for her. And in the interim, she's left a dozen more out in other places that she can reasonably do in that time frame. So drones work, and there are people who use them, and they have a really interesting place where they're helpful, like taking a sample in an area that you can't enter because you're not allowed to take your boat there or walk in. So they are capable of accessing an area we couldn't get to, but they don't take the field work out of the equation, which is what we're trying to do. And they don't work at large scales. And there's permission issues in a lot of places now.
That makes sense. Drone sounds cool. AI always sounds cool, but it doesn't necessarily mean it's the best tool for the situation.
It can be. I saw an amazing demonstration of a drone being used to collect a water sample in a lake that is so protected you're not allowed to go to it, but you could take the water sample from the middle without disturbing the shoreline. That's perfect application of a drone.
Right. Very cool. I have a whole list of other questions, but I see we're already at time. Time has flown today. I'm going to go ahead and get ready to close this out. All righty. Well, I just want to thank you again, Dr. Clare, for that fantastic presentation. I think it took us straight to the end of time. I know I had so many more questions, and I want to thank everyone for joining us today and contributing to the discussion. Also, make sure you go check out the Bio Audio podcast. I know I certainly will be. We hope to see you next week on Wednesday at 11 a.m. Eastern for our 7th and final session in the series, where we will be joined by Dr. Tony Piaggio, who will be presenting on evaluation of environmental DNA as a surveillance tool for the invasive house mice. Second webinar, if this is the second webinar on the potential use of eDNA for monitoring efforts, and the last in the series on environmental monitoring. Once again, we invite you to visit our refreshed Gene Convene Virtual Institute, where you'll find new resources, tools, and publications. And while you're there, please consider subscribing to our free newsletter to stay informed about upcoming events and activities. Thank you, and we look forward to seeing you next time.